I'm here on a job site with Tim, who owns his own electrical contracting business.
3 employees and 2 work trucks.
Tim traded up to Geico Commercial Auto Insurance. We're positively here where he needs us most.
They sure are.
With step-by-step help on all his insurance needs, all for shockingly low rates.
Shockingly low, huh?
Just a little bit of electrician humour. Do you get it?
I got it.
You know, it feels like we have a real connection. All right, I'll stop.
Get a commercial auto insurance quote today at geico.com and see how much you could save. Get more with Geico.
Thanks for selling your car to Carvana. Here's your check.
Whoa, when did I get here?
What do you mean?
I swear it was just moments ago that I accepted a great offer from Carvana online. I must have time traveled to the future.
It was just moments ago. We do same-day pickup. Here's your check for that great offer.
It is the future.
It's the present and just the convenience of Carvana. Sorry to blow your mind.
It's all good. Happens all the time.
Sell your car the convenient way to
Carvana.
Pickup times may vary and fees may apply.
My guest today is Dr. Nirosha Murugan. Dr. Murugan is a Canada Research Chair, Tissue Biophysics, Faculty of Science Distinguished Research Chair, Assistant Professor at Wilfrid Laurier University. Dr. Murugan earned her PhD in Biomolecular Sciences. As an applied biophysicist, her work explores how structured physical signals, such as light, voltage, and magnetism, govern cellular plasticity, tissue regeneration, and the reversal of disease states. Her approach reframes biology not solely as a molecular system, but as a dynamic network governed by first principles in physics. Currently, her lab bridges quantum biology with biomedical engineering to decode how physical signals, acting as carriers of structured information, sculpt the body's energetic architecture. By mapping out how these signals define energy landscapes and reconfigure cellular signaling networks, her work reveals new ways to understand cell state transitions, disease emergence, and therapeutic responsiveness at both the individual and systems level. Through technology, development, and interdisciplinary collaboration, she aims to bring the biophysics of life into real-world innovation that reprogram disease states and support a more precise and holistic and energetically informed model for human health. Today's episode is all about quantum physics and quantum biology. Dr. Murugan will take us through a passionate work and upstream in these biological processes that needs our attention. And now, my conversation with Dr. Nirosha Murugan. I would like to take a moment and mention a pressing issue we are seeing in our modern world. And that is tech use and tech light. The data are impressive and concerning on isolated wavelengths of light, especially shorter wavelengths, which is blue light. For those that don't know, we have chromophores, which are special proteins that absorbs specific wavelengths of light. For blue light, the light that is LED light and tech light, that protein is called melanopsin. Melanopsin is responsible for our circadian biology, physiology and cell functioning, hormone regulation, and even a connection to an area huge for mood. A consideration we should include is this protein was not even discovered until 1998. It seems important for us to understand how our biology uses the different wavelengths of light for various aspects of our biology. This blue light chromophore is our master controller, our master clock. Remember, circadian rhythm is a two-part process, two independent parts: light and dark. For a quick reference, see the 2017 Nobel Prize in Physiology and Medicine. But for now, I have something exciting. I want to introduce a product unlike any other product available. A highlight is the product from Daylight Computer Company created their product based on these factors. The Daylight computer is completely blue light free. It has no flicker. Short wavelength flicker is extremely harmful for our eyes and downstream biology. Light flicker is constantly turning our central nervous system on and off. Essentially, it is like going to a light switch and repeatedly turning it on and off. The problem is, blue light and LED light does this, and it is so rapid you cannot even perceive this in real time. The Daylight computer is the lowest stimulation and foremost for sensory-sensitive users. It is no question that the alternative product, especially when used at night, do not address or consider this in their product. It is so toxic to human biology. Big tech corporations have patents on how their short wavelength implicate the human nervous system. And a bonus, despite Daylight computer not having backlight, it is very functional for outdoor use. And of course, increased sunlight is always preferred. I am happy to offer a discount for the Daylight computer. You can use the code AUTISM for a $25 off coupon. Again, use the code AUTISM and the discount code for $25 off. See the link in the show notes to Daylight Computer Company, or just give it a quick search in your internet browser. Use the code AUTISM for $25 off. I would like to mention Chroma, lights designed for humans. Chroma, a Seattle-based innovator founded by ex-NASA and Air Force engineer Michael Shapiro, is on a mission to enhance physical and mental health with purpose-built devices, unlocking peak human health, cognitive function, and performance. Shapiro launched Chroma to restore the natural light lost to screens in indoor living, and delivers faster recovery, sharper minds, and better sleep. Their products hit hard. The Iron Forge speeds muscle repair with red and near-infrared light, while the Skylight mimics sunlight to boost sleep and energy. A standout, the Forge lamp is a portable gem that fires 660 nanometer and 850 nanometer light to energize mitochondria and heal tissues on the go. Remember the 4 red light chromophores on Cytochrome C Oxidase, frequently talked about in the podcast. This is why. Their products are built with military-grade durability that have lasting impact. Chroma's tech fuses precision and power. Gallium titride power supplies, smaller, cooler, and stronger than silicone, provide flicker-free light that's easy on the eyes. High-powered LEDs target key wavelengths for skin, tissue, and cellular health, with smart heat management for the lasting impact. Every design decision Chroma makes serves a purpose: to create devices that are precise, durable, and effective for improving human life. Remember, humans use different wavelengths of light for different functions of life. Remember when I ask, what do you think light is? Chroma designs with our biology in mind, from sleep aids and wound healing to mitochondrial energy, full body lights and blue light blocking glasses. They are US made and Chroma ships globally and accepts FSA and HSA payments. Use Autism at checkout for a 10% off discount. That's Autism at checkout for a 10% off discount. You just want to start with telling us a little bit about your journey into quantum physics and learning about the biophotons?
For sure. So academically, my interest has always been in trying to understand how biology communicates with each other. He starts in school, in college, and in my undergraduate, I was sitting in an immunology class. And we were trying to learn about antibodies and how they work in the body and how do the white blood cells know where to go, where to target. And we learned about, there are these things called receptors and there's proteins that bind to the receptors, and that's how cells talk to each other. But that didn't sit well with me because if you think about the size of the cell and how many proteins there are, it just didn't make sense that how can this one little protein that's sitting off on one side of the cell, how does it find its way in? So it was an incomplete story for me. And I asked my professor, was there something else? How does a cell know how to communicate with each other? And he said that was it. It was a lock and key model. That's what we knew. And kind of going down this route, I realized that there's more to the cell communication, the cell talk, than just molecules and genes. And that's kind of where this all started, is looking at the physical dimensions of how cells and tissue and biology communicate with each other. So very long-winded answer, but...
Yeah, that's great. And how was your experience with, now correct me if I'm wrong, if I'm mistaken here, but with Michael Levin?
Levin?
Yeah. How was that experience?
Wonderful. When I was finishing my PhD, and my PhD was really grounded in electromagnetic biology and biophotons. We could talk a little bit more about that later. One thing that kept coming up when I was sharing our work was, how does this work? How does magnetic fields affect biology? Like the mechanism, that's always what everyone wants to know. And that the answer took me to Mike Levin because he was the leader in ion channels and bioelectricity. So I thought that had to be the link. If we were going to work with magnetism, we had to think about electricity, bioelectricity. And so that was a link to him. And then working in his lab was the most freeing and one of the most rigorous scientific training that I've gotten. And I'm very grateful for this because it allowed, with Mike, you're able to think very out of the box, very big picture, but grounded in technique and scientific rigor that is required to answer these questions.
Yeah, okay. There's 3 to 5 people that I think are leading the way on this. Yeah, and you two are definitely a part of that. Because I think this is the future of medicine. It's already here. You guys are already doing it.
And medicine's catching up. They're seeing that molecular story is not enough. Everyone's different. Yeah. Everyone's molecular signature is so different. How do we create treatments, right? Yeah. There's more to this than just genes and molecules. I think that's what's easy and for us to measure. The physical side of things is hard to measure. It's hard to train people to think in this domain. I think that's where the challenge is, and I'm excited to see medicine actually shifting this way.
Yeah, it's obviously needed because all the objective data are concerning. There's some few conditions I want to get into later because I think they're definitely in play here. Can you, before we kind of get started, can you explain photoreceptors across the body and especially the opsins? Can you define opsin versus chromophore?
Sure. So photoreceptor, or basically any protein, is light sensitive. Opsins are a class of proteins that are light sensitive and that are able to transduce that information in a signaling cascade. So chromophores are a general category of proteins that are light reactive and light sensitive. So in response to specific wavelengths and intensities of light, they change their conformational position. So they change their shape. And when they change their shape, that causes a change inside of the cell or around the cell for them to kind of understand what's happening.
What does the cell do at that point?
Good question. So typically what happens in signaling is when we think about cellular signaling from a molecular perspective, there are certain molecules that when they change their conformation, they produce other byproducts that kind of in a chain reaction will induce that cascade. Usually this either causes a change in the genome and the genetics that causes expression of genes. They turn them on or turn them off or switches, or they change the metabolism of a cell. So the cell responds to the accumulation of certain proteins. So photoreceptors, for example, when it gets...
I'm here on the job site with Dale, who's a framing contractor.
Hey, good morning.
Dale traded up to Geico Commercial Auto Insurance for all his business vehicles. We're here where he needs us most.
Yep, they sure are.
We make it easy for him to save on all his insurance needs, all in one place, with coverage that fits his business and bottom line. Oh, I shouldn't have looked down.
It's all right.
We're so far up here.
Look at me. Take a deep breath.
Oh, I'm good. So good.
Get a commercial auto insurance quote today at geico.com and see how much you could save. Get more with Geico.
Stimulated by light. They change their conformation, and then inside of the cell, there are proteins that also change their conformation and continue that cascade that will then either tell the cell, okay, we need to grow, we need to stop growing, we need to make more of X protein. That's sort of typical of the cascade.
Okay. Well, yeah, that makes a lot of good sense there. Can you define unstimulated light?
Unstimulated light, is that what you said?
Yeah.
Yeah, unstimulated.
Yeah.
Oh, I think you froze. Did I freeze?
No.
Oh, okay, you froze. Unstimulated light, that's where we call ultraweak photon emission or biophotons. Without light stimulating into the body, these are light that's actually emerging from physics, from energy changes, energy dynamics that exist within the cell. And I can go into a little bit more detail if you want to know.
Yeah, please do. Just expand on that as much as you want.
Yeah, so usually, all proteins, everything that exists in biology, we're all immersed and surrounded by this beautiful sun. So we're always in this bath of light. So proteins, they respond to different wavelengths of light. Biomolecules respond to different wavelengths of light. So that's what we call excitation. When light comes in, that's excitation. Let's say 500 nanometers of light, and then they emit. They'll change their energy states and they'll emit in a different wavelength. We call that emission. Now, some processes, they generate light. So if you take an electron and it's at a certain energy state, when it gets to a high energy and comes back down again, that energy gets transformed into the form of light. So you and I know energy can't be created or destroyed. It just basically can be transformed. So this transformation we see in the form of light. In our bodies that are happening right now, that energy transformation also happens in heat, right? We call that dissipated heat. Now we're seeing that energy can be transformed in the form of light.
Okay. That's like the role, I think, of mitochondria, or is like a dissipative structure.
Mm-hmm.
I definitely want to get into mitochondria in a bit. You want to go into the recent paper from the spring and just tell me about how, like, what you found and the methods of that and everything?
The brain glow paper. Yeah. That's a very exciting paper. It's built on the shoulders and the research that's been happening for the last 50 to 100 years. What we have basically showed in our paper is that the neurons, the brain cells, emit light. And this is not new. People have shown that brain cells do emit light, living cells do emit light. What's exciting about our paper is that we can measure this outside of a living human skull. And that signal of light that comes out of someone's head is actually in relation to or in proportion to their brain activity. So as someone is thinking or doing some cognitive task, that light emission seems to track. And what's really exciting about that is that this is, I think, the first time that we're showing that the light that's coming out of our heads and our bodies is not byproduct. It's not just a random event. It may be instructive. And I want to emphasize the maybe because we need to kind of prove that a little bit more.
Okay. Could you explain just briefly about the photomultiplier? That
Tubes?
device?
For sure.
Yeah.
So the ways to actually pick up the photons, the light is very weak in intensity. It's not as you can't see it with the naked eye. So if you take an LED and you turn it all the way down to the lowest intensity, that's still too bright from the type of light that we measure. So we pick up individual single photons that we can't really perceive, and a photomultiplier tube does that. So the word photomultiplier means it multiplies the photons at the counts. So every time a photon hits the detector, it converts that signal into an electrical count. So every time, for every one photon, you get like one electrical count, and the counter will count every time that hits that detector. And we can multiply that to get a signal.
Wow, that's fascinating.
It's pretty cool. There's lots more cool detectors out there, like CCD cameras, for example, can actually pick up these photons. And there's a group in Japan, Kobayashi, Jaipai Dai. They use these CCD cameras, and you can actually see the biophotons. You can see the changes in photon emissions across people. The difference between what a PMT or a photomultiplier tube can do is that you can get this signal over time. So the resolution may not be that good, but we get really quick dynamics, and that's why it's really good for brain activity. Whereas CCD camera, you can kind of get that spatial distribution.
Is that the group that released a paper around 15 years ago that showed like the human inside the human body is just lighting up?
Yes.
Or is that somebody? Okay,
No,
yeah.
that's it. Yeah, you know your biophotons.
I thought so. I thought that was probably.
Yeah, they've done quite a lot of the groundwork that's building off of other people like Fritz Albert Popp, the Van Wicks. They've all kind of shown this work that cells and tissue, they've done a lot of this work in plants. Bean sprouts is another, like during the development germination, there's a lot of these like highly metabolic activity that happen in plants, and they're easy to kind of use and measure. So they've kind of done a lot of this work that we're building off of.
I'm going to run an analogy across you about germination and in comparison to autism, and maybe I'll just do it now.
Yeah.
So I'm your student right now, so I want you to be a tough professor on me and to just correct me if I'm wrong. So I want to lay out that so artificial light came around, let's say 1890s. Let's just say 1890s. Autism first showed up in 1930s. Johns Hopkins, Leo Kanner, and then in Vienna, Hans Asperger. They were studying this at the same time. This is like 1 to maybe 2 generations after artificial light. So I'm thinking, like, if, as a comparison, if I'm trying to grow a tomato plant, right, and I have the seedling, and I put in the soil, and I have to nurture that up and eventually get it to sunlight. If I don't get the sunlight, it's not going to develop, right? It's going to be underdeveloped. Mm-hmm. And that's what neurodevelopment is. That's what autism is. Mm-hmm. But in comparison, if I give it the proper environment, it has a better chance of developing into an adult plant. Mm-hmm. So my question is, maybe I should have started with the artificial light, but how much of the this energy coming in is responsible for the development of, let's say, a fetal or that plant?
Mm-hmm. I mean, excellent question. And I've been really trying to bring this to light, no pun intended, about the role of the environmental information. And it's not just like pollutants and things like this. The environment itself carries information for our development. The biggest myth is that our internal surfaces are dark. They aren't. There are receptors in places that are within deep brain region, within the fetus, for example, within the womb, that's supposed to be dark, but it is kind of red shifted. There's a glow of red within those areas, and these opsins are within the brain, for example, like there's other proteins and pigments like neuromelanin that are also photosensitive and photoreactive. So there's a big question mark. Why are they there? Evolution doesn't like to waste energy and is pretty efficient. And if it wasn't useful, we would have evolved out of that, right? So to answer the first part of your question, there's definitely some sort of role in there. In terms of light, the light that we absorb from our environment carries energy and information. And the information is not just in the intensity of light, it's not just in the wavelength of light, but in the pattern in which we get that light throughout the day. So, we see this often, for example, in shift workers. In shift workers that have, it's not just circadian, it's not just the fact that they're not going away, it's not their metabolism, it's the exposure to the light, the pattern of light that they're getting. The receptors and the biology is reactive to this. So we don't exactly know what's happening on our internal structure in neurodevelopment, but within plants, for example, we know that plants need sunlight and they need light to grow. And if you pulse the light in very different patterns, you get all sorts of different shapes and growth patterns of these plants. So it's not, you can keep the source of the light the same, but if you change the pattern, you get a different outcome. And I think that's kind of similar to what you're saying, is that it doesn't need to be just that you don't have the right wavelength of light. That's one obvious source, right? I think artificial light misses the spectrum that you need. It misses the intensity of light, misses the wavelength. That's one. The other big thing that's missing is the dynamics of that light, So I think those are things that we need to think about.
Okay, because the mothers of the cancer kids, the mothers of the cancer kids were all professional workers. They were like nurses, professors, school teachers. They worked in these types of environments. 9 of the 11 were college graduates. And, this is 20s to 30s as they're developing into a adult and profession. And the fathers of the cancer kids were very successful people as well, lawyers and doctors, professors, and so forth. So they were all in this type of indoor environment. And then the grandparents of the cancer kids, the 11 cancer kids, they were the same. They were successful. So I'm imagining that when electricity and the artificial light came available, like these families were on it from the get-go. They had the means to equip their house with the so-called modern environment. Mm-hmm. And if we just zoom out now and over time to present day, we can see a shift, and it's pretty similar to removing ourselves from the natural environment and spending more time indoors and with the various different artificial types of light, because that has evolved as well. The rates of autism kind of match that. But in contrast,
can you say a little bit more? I'm interested about that. The rates of autism match what? The lack of light or the introduction of certain artificial lights?
Both.
Okay.
Yeah. But people get hitched on this because autism wasn't an official diagnosis until 1980. So it's kind of easy to compare the rates from the 60s and 70s, but there wasn't a lot of attention on it until kind of probably 1994 in the DSM-4. Yeah, DSM-4. And the definitions have changed, and I'm not discounting other factors being involved, but with the changed definitions of autism, it's kind of opened the floodgates, and it's hard to pinpoint really the Kanner and Asperger type of autism. those two, people want to discriminate between Kanner and Asperger autism, but if you read the Asperger paper, there are footnotes all across the bottom showing that the similarities of the two cases. So, and then regarding the type of light, LED has obviously, and the tech light from the 80s to current has really accelerated, and so has the rates. So there are different things to normalize when comparing rates, and I don't think we do a very good job of that. But it follows so close.
We know, like, when you think about the role of light, sunlight, the first place that people think about is vitamin D, right? Vitamin D synthesis, and that's one big hormone. But the transduction of pigment, like across all human species, we have different pigments on our skin. I might have a little bit more than maybe you, but you still, and everyone still has this transduction of pigments that transduce information and transduce energy within the cellular environment. So I think the role of light, and we kind of can talk about photobiomodulation, where people kind of have harnessed that. They kind of see that certain intensities and wavelengths of light induce very specific effects on cells. That's great. But again, those are all artificial, right? You're picking a very narrow bandwidth of wavelengths, like, with an IR and whatever. But I think environmental light is very important.
Yeah, and that's what I kind of struggle with is like the lux and so forth, the amount of energy from outside to inside. I mean, like on a summer day, there's like 100,000 to 120,000 lux, I believe. And indoor, you're probably only getting a couple of hundred, maybe 1,000. But from what I've checked, it's hard to get 1,000 lux even.
When also we think about inside, the materials that we use, like glass, quartz, we know they're filters, right? They filter out certain wavelengths of light. So you can be sitting out in your sunroom, but you're going to be filtering out certain wavelengths of light. So I think the more that we understand the dynamics of and what the information of each component of light holds for biology, the better we'll understand its role in human health.
Yeah, okay. So going off with that tomato analogy, like because plants have like life stages as well. If you take a healthy adult plant, tomato plant, let's say, and you block the sun from it, it's going to die. It's going to accelerate. It's not going to produce the tomatoes anymore, and it's going to die. And I think that's how we could look at dementia and Alzheimer's things. Also, the rates of those are vastly concerning. Do you see a comparison with that as well?
With the role of like applied light or internal light, because those are very different things.
Yeah, just the loss of energy, the loss of biological energy is the way I think about dementia and Alzheimer's here.
Yeah, I'm with you. I think, similar to that, not so much the loss, but the shift in energy and the patterning of energy. So one study that was published by Jaipai Dai have shown that the wavelength of light that comes from this was done in rodent studies, in young animals versus older animals, the light that's being emitted is redshifted in older animals. So that tells us the energy within those biophotons are different from that of the younger animals. Same neural architecture. Yes, there might be some neural loss, but overall same architecture, but the intensity and the energy within that light is different. So that's one. And then the other thing that we see is that within EEG and electrical patterns, for example, electroencephalography, we can see that the types of electrical firing changes with time. They lose their oscillations. They're a bit more slower. They're a bit more erratic. So they're the physical landscape. So we can see that electrically they're different. We see that the intensity and the energy within the light that's being emitted is very different from young versus the aged population.
Decreased?
Sorry?
Whenever you say it's very different, is it decreased?
Decreased, yes.
Yeah.
It goes from blue to red, so lower energy. Lower energy. Yeah. And then, so there's two there, energy and pattern. Both of them are different.
Okay. So what is it doing to the cell, the overall cell, whenever it shifts from blue to red? How could you explain the cell to me?
So the first kind of stepping backwards is first we kind of need to know where that light is coming from. And that's been the big question. There's a strong hypothesis that the internal light, the unstimulated light, which is what we call biophotons or UPEs, ultraweak photon emission, there's a strong suggestion and strong hypothesis that they come from mitochondria, from mitochondrial activity. So we call them reactive oxygen species. So when cells flux the electrons, they pull off the electrons from the food that you eat into your mitochondria, and they eventually end up in oxygen. That's what helps create the ATP that fuels our body. In that process, we create these things called reactive oxygen species. It's that produces the light. So is when the formation of ROS, you get this change from high energy to low energy that produces these lights. So the couple things happen. ROS are not so good for our cells. They produce damage. They're highly reactive, is a reactive oxygen, which is going to want to pull electrons from all of your cells. So that's what we know. In terms of what this means for our cells, that's something that we're trying to figure out. The big question, I think, what we need to do is kind of blend in the biology of molecules. How do they actually interact with the light? And how does that affect the whole cell as a whole? We see this in photobiomodulation. We see that, certain receptors are sensitive to light based on the intensities and energies, but we don't really understand the whole signaling cascade at all.
Well, that's fascinating too, right? Because melanin helps with the ROS situation, and melanin's obviously absorbing, like especially neuromelanin, with it being such a black substance, is absorbing all of the frequency of light.
Yes, absolutely. There are other proteins within our body that absorb light, right? Like I said, every protein can be excited, right? With enough energy that it can be excited and it can emit light. And that's kind of what we hypothesize is happening in our brain. The biophotons that are being generated from the mitochondria aren't just random. They're used in some form of stimulation or communication with other proteins, but we need to figure out how that happens.
Okay. So is the ROS and this light being created in the mitochondria, or suspected to be created, is that the things like the FO head spinning, and is it like the TCA cycle spinning, catabolic, or maybe even anabolic? And well, that brings up another question: is there different light emission from catabolic to anabolic even?
That's clever. I don't know. I mean, we know that during TCA, the whole point of TCA is to pull the electrons from the carbon that's coming in, right? The carbon source, the glucose in some cases, the proteins in some cases. That's the goal of these steps. Eventually, they end up in the electron transport chain where that electron kind of moves through it to get to its eventual home of oxygen. That oxygen is what's going to help create these reactive oxygen species. So I'm not sure if within the TCA there's going to be much light generation. It's most likely going to be later down the line in that process.
Okay.
Yeah.
With cytochrome C oxidase, because I want to talk about cytochrome C oxidase too with those red light chromophores. From what I know about the mitochondria, there's VDR, vitamin D receptors, which obviously is absorbing strong wavelengths of light, and then the 4 red light chromophores in cytochrome C oxidase. If I'm not mistaken, that's heme A, heme A3, and copper A and copper B. I forget the wavelengths of light, but can you say a little bit more about that interaction?
Yeah, so those two domains are the most light sensitive within the mitochondria, and they're sensitive to red wavelengths, which is why in photobiomodulation, most of the light that you're using is red. 2 reasons, actually. It's because, one, they're longer wavelengths, which means they can kind of penetrate and get into the body. That's one. Two is that, like you mentioned, cytochrome C is heme-based and copper-based, which is reactive receptive to red light within those intensities. But those are things that I'm actually in a collaboration right now where we're trying to measure those things in real time.
Okay, the red light in mitochondria.
What is naturally coming out of mitochondria on their own?
Okay.
Yeah. So if we know what's coming out, then we kind of know what kind of energies are there, and then we can kind of manipulate them and reprogram them as we put them back in.
Like reverse engineer it.
Exactly. Reprogram them.
Yeah. I always think about the oxidative phosphorylation as I want to reverse engineer ATPase, and I think that's a good way of looking at it because you can just kind of find out any deficiency there.
Mm-hmm. And you can kind of go backwards and kind of follow that electron backwards.
Yeah.
I mean, to me, mitochondria are pretty fascinating from an evolutionary and physics point of view because, they were bacteria on their own, and bacteria are magnetoreceptive. Bacteria also respond to light in a very interesting way. So the fact that they're within our cells, some of that information must have been kept.
Okay. Let me ask you about, are these biophotons, and I hope I didn't miss something, are the biophotons interacting with other parts within us? For instance, leptin or the three aromatic amino acids? I guess a better question is the UPEs synthesizing proteins?
Ooh. Again, very little evidence to support this, but theoretically, my hypothesis would be that they wouldn't be because UPEs are pretty low energy, like a single photon that would be emitted from a basically a biophoton would be relatively low energy. You either need a lot of them over time, or you need like a high intensity light generation to induce protein conformational change. It's a lot of energy that's required to break these bonds, right? To kind of break them and change the shape of these proteins. So my guess would be that I don't think they do. I think they might have another signaling role. Maybe it could be like there's work done by Carmen Mothersgill from Hamilton, Ontario in Canada, who's shown that biophotons can act as and induce what we call the bystander effect, where one cell can kind of communicate with each other, where one cell emits the photons and that energy itself can kind of induce another reaction or behavior from a cell. So they see this especially after radiation exposure, where even after the radiation is gone, cells are kind of reacting to each other. And even though there's no information and there's no proteins being shuttled around, there's still light. So they kind of hypothesize that this bystander effect is a communication modality. So to answer your question about whether these biophotons can induce protein level changes, I don't think so. But again, we don't have much evidence for that.
Okay. I've heard that bacteria will synthesize tyrosine and it makes dopamine. But I also want to say, because I wrestle with this, with the aromatic amino acids, like phenylalanine peaks at 260 nanometers, and then like tryptophan and tyrosine is close to 280.
Mm-hmm.
So
the tryptophan is what's really exciting because tryptophan and the interaction with tryptophan and microtubules, for example. Like
I was going to ask you about microtubules for sure.
Yeah, so tubulin itself is also photosensitive, right? It's photosensitive within the UV range, which is similar to tryptophan, right? So tryptophan being also a precursor to melatonin, which is also light sensitive. So there's like a lot of these interactions happening, but
and goes back to the mitochondria.
Correct.
Okay.
No. So the thing to really think about that we need evidence for is what is the wavelength of light, because that wavelength of light will tell us the energetics of that photon. Two, do we have enough intensity or energy to influence biology? perhaps it just takes one photon to induce a signal, but we don't know. We don't know if that is actually enough.
Okay. So I wrestled with. Now I get that TPH1, tryptophan hydroxylase 1, is on the surface in keratinocytes, and I get how that would be synthesized for the tryptophan, but the internal tryptophan and tyrosine and how that is being synthesized, because I don't necessarily think it's just all from food, and maybe you can correct me, but I'm learning that it's not.
What's not from food? The energy conversions?
Yeah, well, yeah, for tyrosine and tryptophan.
Yeah, that I'm not too sure. I haven't really looked into too much of that, so I'm not sure. Again, if the available energy in the system is not enough, there nothing is going to happen, right? So if we're going to say that light is the kind of the conduit for this conversion, there needs to be enough energy within that light, and I'm not sure that the biophotons is that.
Okay, well, that's good information. Can you talk about the microtubules a little bit? What I know, I'll just tell you what I know. So the cytoskeleton is involved, and they're almost like a train track for the cells. But can you just expand on my lack of knowledge there?
Yeah, so the cytoskeleton, as you mentioned, is made up of microtubules. There's actin as well within the scaffold, but for specifically within microtubules, the tubulin proteins form these dimers, so they polymerize, and they basically create like a spiral shape. Where what's really exciting is that the tubulin creates these spirals that kind of creates the rod, but inside of that rod is water. There is what we call coherent water or ordered water within that domain. And this brings up a whole other conversation about the types of energies that this water can hold. And I kind of also want to mention that when we are talking about photons, we're talking about photons, biophotons, all that, but they're still within the electromagnetic spectrum, right? So you're talking about electromagnetic energies at this point. So microtubules and Stuart Hameroff is a strong expert here, have linked this to consciousness and conscious awareness. But the tubulin dimers, they create a form of energetic resonance in terms of the type of light energies that can interact with them. So they are tryptophan, like as you mentioned, is UV leaning in terms of the type of emissions that come out from these proteins. So they're I think from 280 to like low 300 nanometers. So within the UV side of things. Yeah, so there's lots of people have shown that electromagnetic fields can interact with these tubulin proteins, and so basically polymerization is when you take each of these tubulin and they kind of form like a little network, like little Lego blocks that get attached to each other, and when they depolymerize, they kind of loosen. So you can kind of imagine them as like a tightrope walker. If the tightrope is too loose, the proteins on top can't actually move and they kind of fall apart. If they're too tight, they're you risk of over polymerization or damaging them.
Okay, that's a great analogy. This water that you just mentioned with the microtubules, is
Yeah.
that different than the cytochrome C oxidase?
Yeah. So cytochrome C oxidase is specific to, well, it's not always specific, but we're typically in the context of mitochondria. I'm not aware of them being in cytoskeleton or tubulin microtubules.
Okay.
Yeah, it's just tubulin dimers within that space.
Okay. Yeah, I didn't know like where that water is coming from.
It's coming from the environment. So the cell itself it's not dry. It's sitting in water. So the water also, like a small percent of that water is generated from the mitochondria. So
Just a small percent.
yeah, about 4%. Every time a ATP, an energy molecule, is made, it can only be made at the same time of water. So when that electron gets to the oxygen, it pulls in two hydrogens from the intermembrane space and then generates water. So every time you create energy, you are also creating water.
And this is why, like going outside, I think, or going outside, it's like much different energy.
Absolutely.
Yeah,
Different intensities, different wavelengths, all of that.
yeah. So it's so energizing outside, and
Hmm.
you don't even feel like you have to eat as much because of you're stimulating all of that.
Yeah, I mean, and then there's some recent work that I've read that is really exciting about oxygenation, right? Like outside, there's a lot more free oxygen. There's more the breathing rates change, and when we change the breathing rates, you're able to change that electron flux, right? Like how much is
Mm.
actually getting to the cells and how much is actually fluxing through. So, yeah, there's it's a multidimensional process.
I had a PhD or MD PhD. He's like the leading researcher on autism metabolism. He said that the whole reason we breathe is so cytochrome C oxidase can function. And I'm thinking about like being outside, especially in the woods or around a lot of plants, trees, and so forth, because they're putting off um, red light or near infrared light. Like they're admitting that.
And oxygen, right?
Yeah,
They're
yeah, definitely. Yeah.
they're definitely also like absorbing the carbon dioxide that we're expelling and have a more available oxygen for us. So, yeah, there was a paper that I put out there or shared that our brain activities are very different when we are exposed to nature, and it's not just pictures of nature. When you're actually in nature, that you have lower oscillations, less high frequency brain activity, so a lot more calm, a lot more steady state then and I think anyone who's been to woods would feel that way.
Yeah,
So
is it just ranging like at theta or even like up to alpha or
it's
what
alpha. There
alpha?
was more alpha activity produced within the frontal lobes, and those who were walking outside, being around nature. Then, I mean, there's also studies that actually show the same thing in running outside versus running on a treadmill. You are moving
Mhm.
your body, but there's something about being outside, having that free flow of breathing, access to oxygen, less stagnant air, light. All of this plays a role.
Yeah, I'm a big fan of running, and there's a huge difference. Uh,
Hmm.
People get that runner's high, and it's
Yeah,
pretty common.
yeah. I'm the exact opposite. I can't run outside.
Ah,
And this is, I think, a little bit more cognitive. Is because I'm too relaxed outside. I don't feel that urgency, that
mhm,
rush
mhm.
outside. So there's something there.
And that maybe speaks to what you were saying though about the alpha wave and just being calm. Maybe that's it
Yeah,
hits you differently.
yeah. I don't feel that. When you're that excitement to run that you just want
Yeah.
to get going. I don't feel that outside. I see a treadmill and I'm ready to go.
Oh
But
wow.
I outside, I'm envious because my friends who run marathons, they're outside and they can go, and I don't have that urge or energy.
Hmm. It kind of makes me think about locus coeruleus and like the substantia nigra, especially the compacta with that neuromelanin that you mentioned earlier.
Dopamine.
Yeah, yeah, yeah. And the norepinephrine, yeah. Like persisting and getting going.
Yeah,
But
I mean, the substantia nigra as filled with melanin, neuromelanin, for example, as you know, and that is also blue shifted UV leaning in terms of absorption and emission. So there's some structures that are obviously light sensitive in those regions, super, super deep brain structures, but they're
yeah,
still light receptive in there.
yeah, the mesencephalon holds key details into autism, I believe. And I don't know why at neuroepithelial, those four cell types, proencephalon, mesencephalon, and rhombencephalon, and then the fourth one that responsible for the spine. The mesencephalon is so small, but it doesn't further subdivide. Like it's done.
You're talking about the midbrain, like midbrain
Yeah,
pons?
yeah,
Okay.
yeah,
Yeah.
yeah,
Yeah, I mean, those regions are, evolutionary conserved. Those are what kind of gives our basic, or I should say, vital functions, right? The pons, the midbrain kind of help regulate breathing,
yeah. Mhm.
um, cardiac rhythms, things like this. But I'm not too sure of any light specific receptors in those area, so I'm not sure I can comment too much on
Would
them.
it be the? Oh, okay. Like the neuromelanin is what I was thinking.
Okay, yeah, neuromelanin is distributed everywhere.
Yeah,
Um,
yeah.
Yeah.
especially in the midbrain, is what I've learned.
Yes. Yeah.
Oh,
So the raphe nucleus, which produces serotonin,
mhm.
is also red light sensitive. So there
Oh,
might be some link, again, more research. I don't want to give conclusive statements, but
yeah,
because the
yeah,
raphe, because serotonin itself is very light sensitive, and serotonin is a very exciting neurotransmitter to talk about because it's super, very important in neurodevelopment. We see that in early neurodevelopment, if you manipulate bioelectrically these cells to kind of manipulate serotonin 5-HT receptors, you get all sorts of brain development dysfunctions, optic nerve dysfunctions. And again, serotonin, raphe nucleus, 5-HT, all red light sensitive. So there might be a link there to explore.
wow. I want to ask you another like analogy, like because we're talking about nature a little bit too. The trees in the winter will kind of represent how our bodies are, especially at these higher latitudes in the winter.
Mhm.
Meaning we're not doing the biophotons as much. Like we're not lit up inside as much.
I don't know. The data doesn't support that. So,
Oh, really?
yeah, so we've done this. So the paper that we have published, that study was done over a 2-year period. And we haven't published this just yet, but there is no difference in biophoton emission of the same person that was recorded over seasons. So the amount of light coming out of a person, there's no different. So it's not contingent on environment. It's something internal, and we maybe think it's metabolism, maybe it's mitochondria. We don't know yet, but there's no difference between people. So the graduate student that was on this study, we had her record herself every month biophotonically over the year, and there's no real differences.
Okay. Did you track like subjective mood during the season?
So we did do questionnaires related to mood. I would have to look back on that
Okay.
data to kind of, but nothing stands out. The one thing that we're kind of getting ready to put out there is to show that skin type using the Fitzpatrick scale
Mhm.
is a strong correlate of how correlated the brain activity is going to be to the brain activity of light is going to be with the electrical activity. So the more pigmentation, which means you're higher on that Fitzpatrick scale,
Mhm.
the more correlated the light activity to the electrical activity. What does this mean? This is something that we need to think about. I have some ideas. It could suggest that pigmentation across the surface pigmentation that we see has something to do with maybe reflecting that light back in, or maybe involved in kind of transduction. But these are again speculations that we have to validate.
Autistic children are so pale.
Mm.
Yeah, they're so pale. I hope people notice that now because you can't unsee it.
Oh yeah, yeah. I didn't know that. Thank you for
Yeah,
sharing.
yeah. And I think it goes along with this, with that, like the bioelectric and the development.
Yeah, I mean, I know there's like, I think this opens up the door for people to look at cell types from these kinds of signals. Like even bioelectrically, are the cells at a different resting membrane potential than
Mm.
what we call a healthy counterpart? I'm not sure.
Okay. Is there a difference between the amount of biophotons with somebody that's suffering depression versus somebody that's not? Or if it's the same subject whenever they're going through a depressive like episode versus whenever their mood is good?
Great question. This is why we started this study. So our interest was actually to look specifically in a population. Have you heard of chemo brain?
Yeah, yeah, yeah.
Yeah, so these are individuals who are going through anti-cancer therapies, and it's very toxic. It also kills off their brain cells, and they start to develop some of these symptoms of aggression, as for some people, depression, lack of connection, social empathy, things like this. But there's no way of detecting who's going to get it. So we developed the photoencephalography to pick up those neural changes from before they start chemo all the way to the end. And that's kind of like why we developed it.
Hmm.
And we're starting that clinical trial right now, actually. We're kind of halfway in.
Okay.
Yeah.
That's fascinating stuff.
Yeah.
You have the cancer treatment is part of your background, if I'm not mistaken, and a motivation.
Yeah, yeah. I mean, this is kind of where it all started, working with cancer cells. The question that drives me, and it still drives me today, is why doesn't our body or our brains know there's cancer in our body? Like when there's an infection or when you break a bone, you kind of can, like, something's not well here or there. But why doesn't our brain recognize that there's cancer? The obvious answer that people go to is that, cancer is ourself, right? It's our own cells that are going wrong. And that's kind of what motivated it is. Is there like an electromagnetic communication or some sort of biophysical communication that's camouflaged that the brain doesn't recognize anymore? And that's where it was motivated. And so my early work in my PhD, we recorded these biophoton cell signals from cancer cells, and we see that they have a very, very different fingerprint from their healthy counterparts.
That's interesting. The amount of research now on the photobiomodulation and it helping so many diverse conditions is very promising, I think. I'm not saying that's related to cancer, but I know of photobiomodulation studies on autism. Can't wait for that data.
Mm-hmm.
So it's very powerful stuff. I want to ask you, when people are in an energetic discussion, like they're really enjoying each other's company, are the biophotons and even coherence increased?
Oh, great question. So we know this electrically. We know that, that when people vibe, as they say, when people are vibing, there is a brain coherence between two people. They do these studies of hyperscanning two different people at the same time in synced ways. We do see that there's synced coherence in brain activity. Biophotons, to be determined. We just have started this journey and recording light emissions. But that's a great thing to try.
Yeah, it's very fascinating because it's like out of our control. Whenever
Yeah.
we meet, like, let's say a stranger or somebody that we've just kind of started meeting, and sometimes there's a connection there. Like you can have a legitimate connection, and other times it's just like nothing. So I always think about like the quantum biology aspects that could be in play here.
Hmm.
What are some things more recently that you're excited about?
I'm very excited about seeing how much of the molecular signals that we know is actually downstream to the quantum signals, to the electromagnetic and to the photonic. That's kind of my big interest right now. And something that is very apparent, especially because I've done a lot of work within the cancer domain, there are proteins that may or may not be present in certain cancers, but there are light signatures and light frequencies that are apparent in all of them. To me, the dynamics of light and electromagnetism is more of a unifier and more of an easy way to discriminate between healthy and non-healthy cells than proteins are, because proteins, in some people you might express them, some people you may not. And then the other thing is you need to have an accumulation of these proteins to even measure them. So that's really exciting. And then some of the things that we talked about today is these very different experiences. Can we classify these experiences using physics? So one aspect that we're starting to look into is psychedelics. Within the psychedelic community, some subjective experiences that is often talked about is that when they have a psychedelic experience, they see light. It's something that everyone talks about, is that things are brighter, things are clearer, they see light, they see color. What is it about our brain that when we have this altered states of consciousness that, light is the emergent front and center, right? And then, in, like you mentioned, when we think about depression, we think about anxiety, things are darker, things are less vibrant. There's a change in the intensity and the vibrancy of color. So, those are, and I don't know if there's any kind of relationship there to autism in terms of perception of color and light. Those are things that I'm very, very, very interested in looking into.
Yeah, but the whole autistic sensory processing, they have those heightened event-related potentials, especially like early on in the sensory processing.
Yeah, that's more on the exactly on the processing side, but I'm curious more on the
Right.
subjective experience side.
Okay.
Because I know like light sensitivity can induce neural changes, right? Like in the ERPs, like you're talking about vector-related potentials, but the actual subjective experience of light, are they consistent across people with autism? I'm not sure.
Yeah. Okay. That's fascinating. But as I wrote down to make a note here that after I leave like hiking or spending time in the woods, it seems like my visual clarity is increased. Like I can see like things are brighter.
Mm-hmm.
I don't know if other people share that.
Mm-hmm.
From people I've talked to about this, they share it, but
I mean, there could be a couple things that go on. I mean, typically with visual acuity, what you're talking about is a sympathetic response, right? Your sympathetic nervous response. There could be some of that. Again, breathing does this as well. I mean, breath work has become like starting to learn how really strong it is into our physiology. Increasing oxygenation, decreasing carbon dioxide or intake, or having less carbon dioxide in our body. Yeah, I think that those are where those relationships lie. I'm not sure how they relate so much to light. Light can actually maybe support the bioconversion of some of these molecules.
yeah.
But yeah, that would be my guess.
Yeah, that makes sense. Probably within that mitochondria, maybe,
Hmm.
and being the so-called powerhouse of the cell, which is I think they're much more than that.
Absolutely. We're starting to see it. We're starting to measure it. We're starting to see that they're not just powerhouses, that they actually respond to information. They respond to magnetic fields. They respond to light. What I would love to see is if they can hold information. That would be very powerful, is if they're not just like this filter, that they actually integrate that information and kind of have some sort of decision-making process. That would be wild.
So I want to follow up on that. Like each cell or each mitochondria has like a brief summary or a brief memory based off of
The input.
whatever it is. Yeah, yeah. Like because I think maybe cells have a memory, and whenever they're activated, they're passing on through the axon or dendrites or whatever information, and they're just kind of each connection's building a story.
So let me ask you, can I, if you don't mind, I'm very curious about this, and this is kind of, we're getting into the topic here. What is information? So you talked about information going into that axon. Where is it? What is it? Is it the electricity? Is it the pattern into it? And I think when we understand that from what we know in neuroscience, it's the pattern of firing. It's the pattern of electricity that gets through down to the axon and onto the next cell. And if we take that same analogy and kind of apply it to mitochondria, with that electron flow in the electron transport chain, where's the pattern? Is that the pattern that we should be looking at? I'm not sure if anyone's looked into this. And I think those are the types of things that people kind of should reframe in terms of dynamics. So we're not just measuring the number of ATP that may get made. We're not just measuring, how many cytochrome oxidase or NADH gets made. I think we should be looking at the dynamics of these flows. And if once we know the dynamics, then we can actually see that over time. Is that information stagnant, or is that information or dynamics changing over time?
So okay, more efficient things going on within, especially the mitochondrial matrix. All of these things that are feeding into the mitochondrial matrix, there's less bottlenecks.
Mm-hmm.
Is that what you're kind of saying?
I'm saying is like, I want to know where that information is. So if we're saying that the mitos are able to remember, they need to remember something. What is the information that they're remembering? Is it the electron flow? Is it the light that's being made? Is it the turnover of cytochrome C or cytochrome oxidase? Like, what is that information?
Okay.
Yeah.
Confirm or correct this. Whenever I was saying it passes on information, I meant like subjective memory. Like last time I was in this situation, and then all of these cells will start firing and connecting.
A
It's
network.
providing me. Is that what you're saying? That is in regard, or that is created with what you explained?
Yeah, I think so. That's
Yeah,
kind of, I mean, this is a hypothesis. I think so. That would be wild. Um,
yeah,
I think when we start to reframe of like what information is, then we can kind of see if it could be stored, right? And maybe in people with mitochondrial diseases or people who have dysfunctional mitochondria, or even dysfunctional cells, those cells maybe don't seem to remember that information or process that information the right way. And that's what we're calling pathology, right? So that's kind of like a new way of looking at this.
So that's upstream of the subjective, like, valence or whatever that we attach to things.
Correct. Absolutely. Way upstream.
Yeah. Maybe as far upstream as you can go.
I think so. That's what
We know.
exactly. That's where I think modern medicine is like kind of downstream. We're looking at
Yeah, it's very upstream.
the genes, the molecules, the end point,
Yeah.
where with this new idea of like cognitive framing of like memory and learning is a little bit more upstream. And I think way more upstream to that is looking at information. And we can look at that in terms of energetic information. Where is that information? What is it? Can it be stored? And then that kind of brings to the next question. That information, can we learn from it? Can we make decisions from it? And then those decisions lead to us, okay, this is the proteins associated with them. So there's like a little story here. And I'd like modern medicine to move all the way up.
Mhm. Yeah. When the photon hits matter, energy to information.
Oh,
At
yeah,
that moment, that's where we
absolutely.
gotta go.
Yeah, absolutely. And we're starting to see this actually in computation. If we kind of shift gears a bit to computer science, and we start to build all these AI bots, and we're trying to make these computers more efficient. Our brains are pretty efficient at 12 watts of energy. We can hold as much as information as we possibly can. Um, but now we're seeing like in optical biocomputation, we're seeing that the light itself can carry information. I mean, we know this already. Fiber optic cables that run across the ocean for telecommunication, that's information. And I'd like to see that be applied to our body, that there are potential biomaterials that can also carry the optical information, and our cells use it in a meaningful way, integrate it and use it.
It seems like it's very similar.
Mm-hmm.
What's next for you? Like, what's the next thing that, as far as how to find you, and just, I know you mentioned about the things more recently you're excited about, but how can people find your work and look out for these next things?
Yeah, so we have a website where we kind of publish these things, but so the MuruganLab.com. I also have an X account where I kind of try to be active on there. In terms of what we're interested in, the big picture is I want to know where these are, how much information our bodies can kind of carry, our cells can carry, and where that information is. Um, so I look at biology as basically overlapping signals. You have electrical, you have genes, you have this whole stack of signals. And when you collapse them all together, that's communication to me. And we have the first layer of stack of the genes and molecules. We have that dynamics. We get electrical activity. We're missing the top half of like magnetism and light. And I want to see how that plays all together. And I think once we have that, we have a really cool way of understanding our health. And then when shit hits the fan, what we call disease. So that's what really excites me. That's what really drives me right now. And I hope we can make some progress in that area.
Well, this is the way. This is the way forward. I'm glad you're one of the leaders in that, because this isn't going to miss, I don't think.
Yeah. I think there's two things that need to happen is the cognitive framework from our colleagues who are able to kind of integrate what we're saying into their work, because it's not going to happen with just one person, right? So the fact that Michael Hamblin has been talking about photobiomodulation for many years and now it's being accepted, biophotons being talked about, for example, for centuries now being accepted. I think we need, as a community, to do better on looking at the data and integrating it into biology. And then number two is tech. We need tech. If we're going to measure light from very different types of people, we need the tech that's sensitive enough to do that. So working with engineers that can kind of have that language crosstalk between engineering and biology and physics, that's the way to go.
Yeah, back to the Fritz Pop mention.
Yeah.
He wanted that, I think. Well, I appreciate you joining me, and I hope you have a wonderful day.
Thanks, Ryan. I have a question for you, if you don't mind.
Yeah, go ahead.
I typically like to ask this mainly to help connect and help me to understand how we can bring this work forward. What motivates you? What motivated you to think about these very different, non-conventional topics?
So I have many autoimmune problems, and the centralized medical paradigm wasn't doing anything for me. And I thought, well, there's got to be something else. It's missing something. And then in April 2023, I listened to a podcast with Rick Rubin, Andrew Huberman, and Jack Cruz. And Jack Cruz has kind of changed my life with, his quantum biology. And it's worked. It has absolutely worked.
Have you implemented this into your life?
Yeah.
Yeah. And can I ask in what sense?
Increasing sunlight, especially in the morning. I don't miss morning sunlight, and I get UV light. So I have vitiligo, and I used to have really pale skin and used to just wear sunscreen. I haven't had to wear sunscreen since that summer of 2023, and I'm outside all summer. And I don't need it, though. And then I've implemented grounding, and grounding's removed 2 autoimmune problems. Rheumatoid arthritis, which was kind of not very severe, but it was there, and restless leg syndrome.
Okay.
Which is kind of just like a downstream or sequela of the type 1 diabetes, which is another autoimmune problem. But it cleared those out in no time. And the Hashimoto's. Hashimoto's is an autoimmune version of hypothyroidism. And of course, about 15 years ago, I was prescribed levothyroxine, and just over time, my doses increased because that's the only option. And I learned about tyrosine being the aromatic amino acid, and those benzene rings being UV light absorption. And with the pi electrons, I think with the increased sunlight, it rescued my thyroid. I no longer need those, and it's verified twice by different labs at different times. My thyroid levels are perfectly fine without the levothyroxine, so I no longer need that.
Interesting. I mean, I asked this because, a lot of people make these changes when it starts affecting people, and when it affects people, the doctors are willing to listen. So, kind of getting your story out and sharing this and how quantum bio and physical principles can affect the human life, people need to hear that. And so thank you for this platform.
Yeah, and I'll just mention that a centralized doctor, my eye doctor, I went in for an OT or OCT scan, and I was telling him about biophotons, and he basically laughed me out of the room.
Yep, all too familiar with that.
Yeah.
All too familiar. Yeah. But it's the data doesn't lie. If you do rigorous science, the data is there, and it is what it is. So
centralized medicine is a powerful fortress.
Yeah, and we just have to be consistent.
Yeah, you're working to bring that down, and I appreciate your comment earlier about connecting the silos.
Yeah,
I don't like silos.
Yeah, I mean it's in between things where you get information, literally.
Yeah,
So.
The sharing of information, more information talk.
Yeah.
Thanks.